Ok. Bom, gente, a gente vai começar. Bom dia a todos, né? A gente vai começar aqui a apresentar os seminários do Programa Multicêntrico em Bioquímica e Biologia Molecular. E hoje a gente tem o prazer de trazer, né, a professora, a doutora Gabriela Brumatti, cujo trabalho é dedicado a melhorar os resultados, né, para os pacientes com câncer. A pesquisa dela se concentra em caracterizar os mecanismos pelos quais as células cancerosas evitam a morte celular, para promover a formação de tumores e resistência a terapias atuais, né? E também determinar como as vias de morte celular podem ser terapeuticamente alavancadas para a eliminação de células tumorais. Ela completou o doutorado na Universidade de São Paulo, aqui no Brasil, onde ela caracterizou os mecanismos pelos quais os oncogenes regulam a apoptose para promover o câncer. E aqui, então, ela seguiu o interesse em trabalhar com sinalização celular e pesquisa de câncer nos 3 anos de pós-doutorado no Trinity College em Dublin, né? Em 2007, ela se mudou para a Austrália, né, para completar o treinamento de pós-doutorado em duas instituições diferentes. Numa, ela investigou como os fatores de transcrição ROX promovem a formação de leucemias e depois continuou seus estudos sobre vias celulares alternativas e resistência ao tratamento, né? Ela desenvolveu também novos modelos de camundongos e sistemas in vitro, né, de leucemia mieloide aguda, para estudar os mecanismos de oncogene e resistência em doenças hematológicas malignas, né, e também novas estratégias terapêuticas. Atualmente, ela é pesquisadora sênior do Walter e Elisa Hall Institute, enquanto continua a desenvolver o seu programa de pesquisa focado na caracterização da interação das vias de morte celular em células tumorais. Então, hoje ela vai apresentar para a gente um pouco do trabalho dela nessa linha de pesquisa, né? Então, eu queria dar as boas-vindas à professora, a doutora Gabriela, e é um prazer ter você com a gente hoje aqui, né? Apesar do horário para você. Né? Então, eu vou aqui compartilhar aqui a sua apresentação, né? Que tem aqui o título Reaping Cancer Apart, Exploring Cell Death Pathways in Cancer. Então, a doutora Gabriela, do Walter e Elisa Hall Institute. Então, Gabriela, o som está muito baixo. Ok. Melhorou o som, gente? O pessoal está falando que o som está um pouco baixo, mas eu acho que agora melhorou, né? Ok. Bom dia a todos. Então, professora, doutora, é com você.
Obrigada, Jana. Obrigada a todo mundo. Bom, vou começar agradecendo a todos por esse convite e por estar aqui para escutar o meu seminário. É uma situação meio estranha, porque eu estou falando para uma tela com os meus slides, eu não consigo ver vocês, mas eu espero que vocês consigam me ver um pouquinho, ver a apresentação e me escutar bem. Eu vou começar também pedindo desculpa, eu vou dar esse seminário em inglês, porque faz um tempo que eu não apresento resultados científicos em português, e aqui são meia-noite. Se eu fizer meu cérebro tentar apresentar em português, não vai ser uma coisa muito legal. Mas eu estou à disposição para vocês fazerem as perguntas em português e eu vou respondê-las em português. Mas o seminário será em inglês, tá? Então, bom, vamos começar, né? So I'm talking about reaping cancer apart, so exploring cell death pathways in cancer. So as Jana said, I've been working on cell death pathways for a while, and my interest was always to try and understand how oncogenes regulate the cell pathways to promote cancer, but also understand how once the cancer is established, how this cell death pathways are deregulated for the cancer cells to evade cell death and also resist to chemoresistance. So I've been working at the Walter Eliza Hall Institute now for the past 10 years, and that's the main work that I'm going to present here was building this institute. So this is just to start off with the hallmarks of cancer. I think most of you must have seen this. This was published by Hanne Weinberg in 2000 and then 2011. And there are several pathways that are extremely important for the establishment of a tumor. And one of them is the resistance to cell death. And that's what my research has been concentrating in, and I'm going to talk to you about it. So if we think of cancers and cell death, there are two things that we think about it: the promotion of tumors, so tumorigenesis, and the treatment resistance. So those are the two main topics of the cancer that are really regulated by cell death. And the cell death that comes up to our minds is always apoptosis. Apoptosis is a caspase-dependent cell death, anti-inflammatory. So as several of you may know, there are two main pathways to induce apoptosis. There is the intrinsic pathways, where cell death stimulus go through the mitochondria to then form the apoptosome and activate caspases that then will lead to DNA fragmentation and cleavage of downstream substrates that will lead to cell death. Or there's the death receptor pathway, in which ligands connect to their death receptors in the membrane of the cells to activate caspases and then leading to cell death. These two pathways may be interconnected, especially with the death receptor pathway, where pro-apoptotic molecules such as deadh can then activate events on the mitochondria that will also lead to cell death. But the most important thing about apoptosis is that a very silent type of cell death. We have these apoptotic bodies and changes on the surface of the cell that make this cell being quickly eliminated by macrophages and other immunocells in the system, which does not allow an inflammatory process to occur. So most of the chemotherapies and treatments that are there are to target these death pathways. And that's mainly because it has been shown that members of the BCL2 family that control apoptosis or inhibit apoptosis are upregulated in tumors. And apoptosis has been very connected to cancer. The problem is, and actually the deficiency of the anti-cancer therapy since all these studies started has been amazing. So if we look at the progression and survival of patients since 1975 until 2015, these are three different types of leukemia. And we see that now, especially patients with CML, where Gleevec and kinases inhibitors have been elaborated now, have a much better prognosis and chances of survival. The problem is despite of all these new treatments and the studies that have been done in the field, there are tumors such as acute myeloid leukemia, which the survival rate still remains really low. So acute myeloid leukemia is one of those tumors that are very aggressive and very hard to treat, mainly because it's a heterogeneous tumor and mainly because they're extremely resistant to chemotherapy. So the overall survival past 5 years is less than 30%. And even if a patient survives the first initial treatment, they tend to relapse, just indicating that this kind of cancer can invade and build resistance to the treatments that are out there, and the disease comes back. So why do the anti-cancer drugs fail to improve AML survival or the survival of other hard-to-treat cancers? So there are a couple of things. One of the things is the mechanisms of the resistance. So if we try to understand this mechanism of resistance, we have a better chance to improve the outcome for these patients. But the other things that we have to keep in mind is explore new therapeutic strategies. So In my research, we are mainly focusing two different ways. So we are looking to targeting non-apoptotic pathways. As I said, apoptosis is the pathway that has been mostly studied and most approached to treat cancers. And we are now, with the evolution and understanding of new cell death pathways, there's a possibility that targeting different cell death pathways might be a way to overcome apoptosis resistance and also identifying new targets. But what I want to focus today on my talk, because of the time and because of the topic, is to how we are going about targeting non-apoptotic pathways. So the non-apoptotic pathway that we are focused on is necroptosis. This is a cell death that has been discovered not too long ago, about 10 years ago. And it's a very different type of cell death. First of all, it's a caspase-independent cell death. So it does not share the same components that apoptosis require to kill a cell, which is very beneficial if you're thinking of targeting a different pathway to kill cells that are resistant to apoptosis. But in the second instance, what is important to know about necroptosis is that it's an inflammatory type of cell death, which means that when you induce this type of cell death, you also activate the immune system. And in several cancers, this is quite beneficial because you have then a much stronger anti-tumor effect from the system around the cancer cells, and you're not only targeting the cancer cell. So just in a briefly description of what is the necroptosis pathway consists of. So the most described signaling pathway of necroptosis is the one involving TNF and TNF-1. So when TNF binds to the TNF-1 receptor, what it happens is, in the absence of caspase-8, which can be by addition of a pharmaceutical caspase-8 inhibitor or by the overexpression of endogenous caspase inhibitors such as FLIP, we have activation of RIPK1 that then connects to RIPK3, phosphorylates MLKL. MLKL goes into the membrane of the cells, forming pores, and now we have release of the DAMPs, which are cytokines and contents, intracellular contents to the extracellular environment, leading to an inflammatory response. So this is just a schematic of really comparing apoptosis and necroptosis. While in apoptosis, we have a membrane pretty much intact along the way until the cells die and are recognized by macrophages. Necroptosis is a mix between a necrotic cell where the membrane is compromised, but it is a regulatory type of cell death where we can still control and still have key components happening. So our lab was working quite intensively in the necroptosis signaling, and we coming along with several publications that were out there suggesting that necroptosis can be connected to cancer. We decided then to explore that more in our model systems. So, for example, mutations in the RIP kinases that are crucial for induction of necroptosis and MLKL were identified in human cancers, such as the leukemias that I've been talking about, but also solid cancers. Expression of necroptosis-related genes in cancer have been shown to be downregulated or upregulated. So it's still a very area of discussion and of research, whether necroptosis work as a tumor suppressor or a tumor promoter. And we can discuss that after my talk, because we also have been working on that matter, but also has an antimetastatic response. So it regulates NKT function. So deletion of the RIPK3 kinase, which is important for necroptosis affects NKT activation and leads to antimetastatic response, and the immunogenic cell death, as I was saying. So the release of DAMPs, the damage-associated molecular patterns, leads to an inflammatory process and activation of the immune system, which can contribute to the elimination of the tumor. So the idea was, can we use necroptosis now to treat those cancers that are highly resistant to apoptosis and now kill those cells in a different manner that does not require components of the apoptotic pathway? So at the time, we were very fortunate to also be working in the lab with a class of drugs, the SMAC mimetic drugs. So the SMAC mimetics, they are drugs that were built to target the inhibitors of apoptosis proteins. Those proteins are known to be inhibitors of apoptosis, so they were described to inhibit activation of caspases. And these SMAC mimetics, they were built to bind and degrade those IAP molecules. So why were they generated? So there was a strong evidence in the literature that the group of IAP molecules were upregulated in cancer. So in AML, colorectal cancer, and so on. And in all those cancers, there has been an association with IAP levels and poor survival and unfavorable response to treatment, suggesting that the high expression of these anti-apoptotic molecules will lead to resistance to cell death. So SMAC mimetics were built to target these proteins. And how does exactly this drug work? So in a normal situation, you have TNF binding to TNF1, and if you have IAPs, you will have a signaling process leading to NF-kappa B activation and pro-survival signal. If we now come with a SMAC mimetic, here in our study, we use birinipan, that is one of the most advanced SMAC mimetic in clinical trials. What happened is there's a degradation of IAPs and now activation of the cell death, apoptosis cell death pathway with activation of caspase 8. And what is important with this drug is also, at the same time that induces cell death, it promotes production of TNF that we don't really know exactly how it's working, but induces TNF, and then this pathway can feed on its own and keep activating the cell death in the cells. But what is very interesting about this drug is that not only induce apoptosis, but we can also induce necroptosis with it. So in our systems, if we use caspase inhibitors or caspase 8 inhibitors especially, we now have a formation of this complex 2B with RIPK1 and RIPK3, induction of phosphorylation of MLK and necroptosis. So if instead of just treating the cells with birinipan, we treat the cells with birinipan plus a caspase inhibitor, we block apoptosis, but we induce necroptosis. And that's what is going to be the focus of my study today. So as I mentioned, AML is one of these type of cancers that is really hard to treat and has a need to new therapeutic strategies to be developed and discovered. So we just develop in the lab model systems to understand how these tumors develop in terms of cell death signaling, but also test new therapeutic approaches. So the way we develop these models in mouse, we take progenitor cells from E14 livers, and we transduce them with oncogenes. So we infect them with viral containing specific oncogenes that are associated with the development of the disease in humans. And then we inject those cells into irradiated mice. And then after a while, these mice would develop the leukemia. We are able to harvest the cells from the bone marrow and spleen, and we can then perform molecular cell death analysis, but we can also, and that's the important thing, look at the efficiency of a treatment in vivo. And MLL-AML, as I said, it's a very heterogeneous disease. It has different genetic mutations that are associated with the disease, but essentially there is a group of these mutations, the MLL translocation oncogenes, that are the ones that have the worst prognosis if a patient presents with these translocations, and they are highly resistant to treatments. So there is 8 MLL translocations that have been identified in patients, and we were able to set up in the lab 5 out of those 8 translocations in mouse models. But here's also a list of other models that we are working in the lab, and they are also associated not only with the MLL translocations, but with also mutations in RAS, and HOXA9 is another transcription factor that has been strongly involved in the development and establishment of leukemias in patients. So we have at the moment 15 different models, and we can test all our treatments and our hypotheses in all these models. And you can see there is a those models vary on their time length as well. So what we decided to do then, we took cells from different models, here 4 different models of our murine primary AML, and treated with birinapant initially to see how these leukemias responded. So essentially they are all acute myeloid leukemias, but you can see that depending on the oncogene that they are driven by, the response to birinapant is very different. And we noticed that some of these leukemia are highly resistant to birinapant. And if we look at primary patient samples, we also see the same pattern. There is a very different response between the different patients that are highly resistant to birinapant, here highlighted in red and dark green, but also patients that are highly sensitive to birinapant. So we decided to concentrate on these patients that are highly resistant and these leukemias from mouse that are highly resistant to birinapant, and see if we could overcome this resistance by combining birinapant with caspase inhibitor. So I think the caspase inhibitor that most people have used in research and in the lab so far is QVG, which is known to be a caspase 3 inhibitor, which is the downstream caspase, and ZVAD, which is a pan-caspase inhibitor, which means that inhibits most of the caspase. And what we did, we treated MV411 cells, which are human AML cell line that are highly resistant to birinapant, here highlighted in this column. If we treat them with the caspase inhibitor QVG, we have no difference. If we treat birinapant and QVG, because QVG is mostly a caspase 3 inhibitor, we don't see much of a difference. But if now we treat with birinapant and ZVAD, we block caspase 3, we block caspase 8, but we now are able to increase cell death. And the pathway that we think we are activating here is a necroptotic pathway because we don't have caspase. The reason why ZVAD seems to be more potent than QVG is because ZVAD is probably a better caspase inhibitor than QVG. That was all great. The problem is QVG and ZVAD are not really drugs that we can think of taking to the clinics, or drugs that are really good testing in vivo models because they have a toxicity on their own in vivo. So we were searching for a caspase inhibitor that we could translate closely to what could be possibly a treatment for patients. And we came across this caspase inhibitor, IDUM we call, but Americasan is the commercial name, but it's called IADN 6556. So this caspase inhibitor seems to have a very potent activity in inhibiting caspases, in especially chronic hepatitis and in liver damage situations. And when we were starting to work with this caspase inhibitor, 2 years later, this drug has been FDA approved in America. So it is a drug that we could not only use in our in vivo systems because the profile had been established, but also is a drug that we could think of translating further down the track. So what exactly is thought about the caspases and leading to necroptosis? So the literature suggests that there is a heterodimer forming of caspase 8 and FLIP that blocks necroptosis. If we come with caspase inhibitors that are very potent to inhibit caspase 8, this heterodimer is broken, and now we can induce necroptosis. So if we did an analysis between the caspase inhibitors that are mostly used in the lab, QVD and ZVAD, and compared to IDUN 6556, and what we see is that the capacity of IDUN to block caspase 8 and disrupt this heterodimer is much stronger than ZVAD. Here, QVD seems to appear stronger, but the ratio is still quite high. So we did a test and compare the caspase inhibitors with the drug birinapant, with the SMAC mimetic, and see their capacity to induce necroptosis in one of our murine leukemias. And focusing here on the full symbols, so the round for QVD, the triangle for ZVAD, and the square for IDUN, you can see that at a very low concentrations, IDUN already induces a high cell death when in combination with birinapant when compared to QVD or even ZVAD. Eventually, ZVAD at a higher concentrations gets very close to the activity for IDUN, but IDUN seems to be a much more potent caspase inhibitor to induce necroptosis. So having that in mind, we went on and tried to characterize the type of cell death that IDUN or amikazan was inducing with birinapant in our cells to confirm that was indeed necroptosis. So necroptosis, just a reminder, is a cell death that in the absence of caspase 8, we have RIPK1 and RIPK3 forming a complex and inducing phosphorylation of MLKL. So the key components of these cell death pathways are the TNF and TNF1 ligation, and also the RIP kinases and MLKL. So we were able to generate leukemias out of mice lacking the specific components of this pathway, and we did short-term survival assays and long-term survival assays with these leukemias. So if we focus on birinapant, you can see that most of these leukemias, this is a very low dose of birinapant, don't die with birinapant alone within 18 hours of treatment. But if we combine birinapant plus IDUN in a wild type, we pretty much eliminate this in a short-term assay. And when you look at a clonogenic assay, we can no longer see colonies, suggesting that that's not only a very strong death stimuli, but most likely, if you would think of treating a cancer with this, the chances of a cancer surviving and the disease relapsing is much lower. So these assays are 14 days after the cells have been treated. When we delete TNF1, so upstream on the cell death signaling, now we rescue these cells from necroptosis cell death, and that can also be observed in the long-term survival assays where the same number of colonies are forming as the controls. Now, what was interesting for us is when we look at RIPK3 knockout cells, we would expect that our cell death would be completely rescued. In a short term, they seem to be quite rescued. They are not as dead as the controls. But in the long term, we do not see formation of our colonies any longer. And the same happened for MLKL. In a bit less effect, in the short term, MLKL rescues the cell death, but in the long term, we could not see colonies. So it means that in the short term, RIPK1 and MLKL are able to rescue this phenotype, suggesting that necroptosis is happening. But in the long term, it suggests that maybe other cell death pathways are being activated. So to investigate this further, we did a time course analysis of cell death in wild-type MLKL and RIPK3 cells, and what we observe is until 10 hours of treatment, while the wild-type cells are over 60% death, our MLKL and RIPK3 had shown no death. After 10 hours, RIPK3 and MLKL start to die, and wild type, of course, are nearly gone. So after 10 hours suggests that there is another pathway occurring that is no longer necroptosis that is leading to cell death. So our hypothesis at the time was that IDUM, it's a great caspase inhibitor, but it has a short half-life. So could it be that even though we have IDUM in the system, the effect of IDUM is no longer really active to inhibit caspase, and now these cells are now dying by apoptosis. So what we decided to do, we did the treatment birinapant plus IDUM, and here again we see in wild type, they are completely dead. In MLKL, there's a partial rescue, as same as RIPK3. But then what we did is that we treated with birinapant plus IDUM, and after 6 hours, we added more IDUM to make sure that there was enough drug in the system to inhibit caspases. So with the wild type cells, we didn't see any difference as we would expect because those cells are pretty much dead. But in the MLK and in RIPK3 now, we almost had a full rescue of the cells, suggesting that now these MLK and RIPK3 knockouts were also dying after a while by apoptosis. So this indicated to us that a birinapant plus IDUM treatment not only was able to kill cells by necroptosis very quickly in the beginning, but was also able after a while to continue killing cells by apoptosis. So we would have two pathways being activated simultaneous or one after the other. And that could be a very beneficial in a cancer system where we have a very large number of proliferating cells, but also heterogeneity between the tumors. So in looking at that, then we wanted to confirm that this combination was very effective in eliminating different models of AML cells. So first we look at cells that were intrinsically resistant to birinapant, which is marked here in white. Sorry, the legend was cut on the slides, but the white are representing birinapant. And when we treat then these cells with birinapant plus IDUM, we could completely eliminate these cells. And this is a human AML cell line that has already a intrinsic resistance to birinapant or to SMAC mimetic inducing apoptosis. But we also wanted to assess if this combination would be effective in a model where you have acquired resistance. So SMAC mimetics are in clinical trials, but they fail because as a single therapy, they were not good enough to eliminate cancer cells. And one of the hypotheses that eventually patients acquire resistance after a while to this drug. So we stimulated AML cell lines from mouse the same way, so with increased concentrations of birinapant for 4 weeks to simulate this process of acquiring resistance after treatment. And then we sorted for PI negative cells, which means we are sorting for survival cells, and now we call MLL, ENL resistant leukemias. So we wanted to know if our necroptosis therapeutic could now overcome this resistance that the cells acquire. So if you look at sensitive cells versus resistant cells, when we treat them with birinapant, we can see our resistant cells are highly resistant to birinapant. In the presence of IDUM only, we don't have any difference. But in the presence of birinapant and IDUM, this drug combination can now convert the resistant cells to a sensitive cells to birinapant, suggesting that necroptosis is indeed able to overcome apoptosis resistance. Another important thing for us was to assess whether this combination could be a way to overcome chemotherapy resistance. So as I said, AML is a disease with high incidence of relapse. And one of the reasons is that these patients, they acquire resistance to the chemotherapeutics that are used at the moment. And one of them is the cytarabine, or RSC, as we call. So we generated in the lab a leukemia that is now RSC resistant, so it's unable to be killed by the normal chemotherapeutic used in the clinics to treat these patients. So you can see here at increased concentrations of this drug, these cells cannot be killed. If we treat them with birinapant, we see a slightly increase of cell death, but it's still 50% of these cells are able to survive the treatment. But now if we induce necroptosis, we almost completely eliminate this leukemia that was highly resistant to apoptosis. So once again, showing to us that necroptosis is able to overcome apoptosis and may be a way for us to look into alternative treatments for those patients that have no chances of recover after normal anti-cancer therapies. But that's all good, and we showed a lot in vitro, but the most important thing is also to know if this is a feasible treatment in vivo. And one of the important things is to know if this is, how toxic is this combination to normal cells. So we did this analysis in human, human cells, but in also mouse healthy cells. So just focusing on the human cells, we took CD34 positive cells, which are like stem cells, like from patients, and we treat the cells with birinapant in the absence or presence of IDUM, compared to cytarabine and daunorubicin, which are the standard of care chemotherapies used to treat AML patients. And what we saw is that, as we know, cytarabine and daunorubicin are extremely aggressive chemotherapies that induce cell death of the healthy cells as well, and that's why those patients are very susceptible to infections and are immunosuppressed during the treatment because all their cells are pretty much compromised with those treatments. But when we treat now with birinapant or birinapant in the presence of IDUM, those cells doesn't seem to be compromised as much as the chemotherapy, suggesting that this is even a better way of treating those patients than the chemotherapeutic that are out there. And the same was observed when we look at different subsets of cells from mouse. So if we compare birinapant plus IDUM with the standard of care chemotherapies, we see that birinapant plus IDUM does not induce as much cell death as the chemotherapies, especially related to B and T cells. So that was very promising to us. Now, what we needed to know, is this really effective in vivo? Because even though IDUM had been tested and had been approved in FDA, and the SMAC mimetic birinapant were in clinical trials, never the combination of the two had been proven in an in vivo system to really promote cell death. And we were not sure also how necroptosis would be in vivo, because would be expected to induce a very strong inflammation, and that's also can be beneficial but can be detrimental for the system. So we set up our in vivo system where we have tagged to our leukemia a luciferase gene, where we can monitor the progression of the tumor with imagings of the tumors. So here, just looking at vehicle, you can see that within 25 days of injection of tumor cells, most of the mouse is compromised and has the leukemia. When we treat those with iDoom, we still have the mice highly susceptible to a leukemia progression. With birinipan, it seems to have a delay, but eventually those mice succumb to the disease. What was interesting to see is that when we then did necroptosis, actually we could have these mice living for much longer without any tumor. And here is just the survival curve that we can observe. So in general, most of our mice die within 25 days when they are in wild type or iDoom treated. Birinipan prolongs a bit their survival. And now we have around 45 days of survival. But with iDoom, the 50% mark is up to 75 days. So we pretty much tripled the time frame of these leukemias. But eventually we had some mice coming with some tumors. Interesting enough, they were not blood tumors. They were tumors that were localized in other areas of the mice. And we don't know exactly what was it, because it was a couple of mice and not all of them. But that was quite a striking result, because these models in vivo of this type of leukemia have very little, the drugs that are tested nowadays have very little benefit if you look in these in vivo models, because they're quite aggressive. So having from 25 to 75 days was a massive achievement and results for the treatment of the disease. And here is just a histology showing of how our treatment is being effective. So if you look at the spleen and liver of the cells, the liver, it's more pronounced. You can see infiltration, the purple ones are infiltration of leukemic cells in the liver. The spleen, these are the splenic follicles, and you can see less of them and more empty spaces, or actually full spaces of leukemic cells. With iDoom is the same. With birinipan, you still get infiltration of the cells, and the splenic follicles, there's still a lot of gaps, so infiltration of leukemic cells between the follicles. But when you look at birinipan plus iDoom, you can see there's many more follicles than empty spaces in between, suggesting there is almost no infiltration, or actually in this case, there's no infiltration of leukemic cells, and also the liver was pretty clear. And that was quite striking to see with this treatment. So that was very promising and exciting for us. So we decided to then go, of course, to patient samples and see how this treatment was effective in patient samples. And this is just a snapshot of some of the cells we tested. So we tested some of AML patients, the AML5 and AML18, and you can see they were highly resistant to birinipan. But when we treat them with birinipan plus iDoom, we could sensitize the cells to cell death. But it was also interesting because you tested ALL, so acute lymphocytic leukemia, another type of leukemia. The one that was highly sensitive, resistant to birinipan, could also be sensitized by necroptosis. So this data suggests that not only necroptosis could be applied for the treatment of AMLs, but also ALLs. So just to summarize what I spoke so far, what I hope I show you was that birinipan plus emirkazan can induce necroptosis to kill AML, that this is efficient to kill birinipan and chemotherapy-resistant leukemias. This is a drug combination that is well tolerated in vivo, but also can reduce tumor burden and prolong survival. And overall, this suggests that the combination of IAP inhibitors with caspase inhibitors may be an effective strategy for the treatment of apoptotic-resistant tumors such as AML. But like everything in science, things are not always black and white, as we would hope. So when we look at a bigger cohort of patients, and here we have 8 different patients, we treat them with birinapant, and again, we saw very response in these patients related to birinapant. When we combine with IDUM, we saw that 3 out of the 8 patients responded really well to this combination treatment, and there was an increase in cell death. But now we had also patients that did not respond to the caspase-8 inhibition, or patients that actually acquire resistance to the treatment, suggesting that perhaps in these orange mark patients, these patients are now dying mainly by the cells of these leukemic cells are mainly dying by apoptosis, and the inhibition of caspases is not inducing necroptosis. If that's because those patients do not have key components of the necroptotic pathways, we still don't know. So in trying to understand that, we are working at the moment with 2 model systems to understand the mechanism of necroptosis resistance using AML as our model. One is a target approach. So we have these KG1 cells, which are another human leukemic cells. We see they are very resistant to birinapant. When we treat them with this other drug that has a target of one of birinapant resistant genes, which is the multi-drug resistant gene, we can now sensitize the cells to birinapant. But if we treat the cells with birinapant plus IDUM, the cells do not die by necroptosis. When we look the cells compared to other cells like the MV411 that we know can die by necroptosis, and look for all the components of the IP families, but also of necroptosis signaling, what we observe is that it seems like there is a reduction on the levels of RIPK3 and perhaps MLKL on the cells. So what we are working at the moment, we are re-express human RIPK3 and human MLKL in these KG1 cells, and then seeing if the re-expression of these genes now sensitize KG1 to necroptosis. And if they do, then we will look at the mutation analysis and understand why the cells are now resistant, and see if there is specific readouts of RIPK3 that can be indicated as biomarkers of resistance to necroptosis. Another system that we are using is an unbiased approach, where we come with necroptosis sensitive cells, such as the human AML cells that I showed you before, MV411, and one of our mouse leukemias, MLLAF9. They are both sensitive to necroptosis. What we are doing with them, we are introducing a guide RNA, CRISPR library into the cells, and once we have the cells infected with this library, we induce necroptosis, and we look for cells that are able to survive necroptosis. So essentially, we generated a necroptosis-resistant leukemia. And once we have those, we are able to sequence those leukemias and identify perhaps mutations and target genes that are lacking on these leukemias that can be an indication of how necroptosis can be inhibited. So far, when we treat those cells, what we saw is that we have here in purple, we have birinapant with IDUM and TNF to make sure that we are really induced cell death, and these cells are dying. When we have now this clone 1 and clone 3, that we put the CRISPR library, now we see the cells are resistant. So now we have a few clones that are resistant to necroptosis, and we are working on those clones and looking at the mutation profile of those clones to look for markers of resistance for the necroptotic pathway in cancer. So just in conclusion with what I spoke so far, my research focuses cell death signaling in cancer. We are looking at possible target therapies. We I haven't shown, but we did look, we have projects working on mechanism of resistance for specific drugs that are in clinical trials and did not respond the way we would hope. And with those identified biomarkers of response to make it therapies more efficacious and promising for the outcomes for patients, looking for new targets, understanding how tumors are developing and seeing if we can identify targets that have not yet been therapeutic explored, and see if those can be now used as a new therapeutic strategy. And what I show you today, which was the alternative cell death pathways, necroptosis being one of them, but of course there are other types of cell death that we are looking into explore and identify as potential therapies. What I would like to emphasize is mainly what we try to do is work with compounds and functions that are rapid translated to the clinic. So all the compounds that I mentioned in here in my talk are already FDA approved or in clinical trials, which makes the pathway to clinical trials much shorter. So if we can provide strong evidence that the combination of specific compounds can be as strategies for new therapeutic approaches that may lead to quicker outcomes for patients. So I just want to end by acknowledging and thanking you all for hearing my talk, but mainly from the people that have been involved on this talk, John, Silk, and David Vo, which were my main mentors and which labs I have been working for the past 5 to 6 years, and they have been amazing, supportive, and give me the independency to develop these projects. Najua, which is a postdoc in the lab, which we have a really good and not only friendship, but a strong collaboration and in developing all those projects. At the time, my PhD was Chunya Ma, that has developed most of the work I spoke about it today, but I have other people that have been working with me, students and research assistants that have contributed immensely to this work, people that contributed with analysis of the bloods of the mice and patients, and also helped me with the model systems. We have an amazing program at WeHi in which we are paired with consumers, we call, which are patients or families that have been affected by the disease we work, and we get a perspective from their view of what is important to research. And I've been very fortunate to have them involved on the project, people at other institutes at WeHi, but also in America that have been crucial for the development of this work, all the staff that work in the animal facility at the institute, and all the funding agencies that have supported me through these years. And thank you for your attention. Jana?
Estou aqui. Estou ok. Deixa eu ver aqui. Eu entrei. Tá. Tá me ouvindo aqui?
Eu estou te ouvindo, mas não estou te vendo.
Ah, tá. Agora você consegue me ver? Ah, tá.
Acho que não. Acho que eu tenho que dar um end show or stop sharing.
Provavelmente. Eu vou remover aqui. Tá? Porque talvez se você precisar de alguma coisa da apresentação, eu posso colocar de novo.
Agora eu te vejo.
Tá?
Agora eu te vi.
Está ótimo. Bom, gente, queria agradecer a doutora Gabriela e saber se alguém tem alguma pergunta. Eu tenho várias perguntas, inclusive. Eu vou selecionar só 3, porque eu fiquei realmente muito curiosa com algumas coisas e eu preciso me conter, mas eu tenho muita pergunta. Então, eu queria saber se alguém tem alguma pergunta. Estou observando aqui. Bom, enquanto isso, eu vou fazendo as minhas perguntas, né? Porque eu trabalho com a apoptose já há muito tempo, né? Desde sempre, desde o meu doutorado, desde 2001. E aí eu vi o surgimento dessa discussão sobre a necroptose tem um tempo, né? Inclusive o Ricardo Vines também publicou alguma coisa sobre isso, acho que foi uma revisão, se eu não me engano. E aí eu queria fazer umas perguntas, 3, eu selecionei 3 aqui que eu achei Então, é importante que no meio da sua fala, você falou sobre a questão da resistência dos pacientes. E aí depois eu vi que você vai começar um trabalho ainda para identificar quais seriam esses fatores de resistência. Então, a primeira pergunta, né, até porque eu tenho meus alunos de pós-graduação assistindo aí também, qual é a principal diferença em termos dessas vias de necroptose para a necrose propriamente dita?
Bom, a necrose não é uma morte celular organizada ou molecularmente controlada. Então, a necrose acontece mais rapidamente que a necroptose e é um processo aleatório, né? Que a célula explode e morre e forma toda aquela bagunça no sistema. A necroptose é um sistema controlado, tem componentes críticos. Então, se você tem uma necroptose celular e você tira MLKL ou RIPK3, o que são as moléculas principais dessa via, você vai bloquear a necroptose, né? Então, atualmente, já tem companhias trabalhando em inibidores específicos da necroptose e os targets são RIPK3 e MLKL. Então, para necroptoses, você pode controlar, você pode inibir. Necroses, você não tem um target específico para essa inibição. Então, essa seria a principal diferença entre elas. Mas é por isso que é uma necroptose, porque é uma combinação entre a apoptose, que é uma morte controlada, e a necrose, porque tem esse fator da membrana se fragmentar e ter toda essa extravação do conteúdo intracelular e inflamatório, processo inflamatório.
Muito bom. Qual era a outra pergunta aqui? Ah, sim. Essas bombas, eu não sei, eu não conheço essas leucemias, né? Que eu trabalho muito com a mieloide crônica e tal. E tem alguma coisa com relação à superexpressão de bombas, essas substâncias, né? Todas essas que já estão aprovadas pelo FDA, elas são substrato para essas bombas de fluxo? Essa leucemia, ela tem superexpressão dessas bombas?
Então, esse é um artigo que a gente acabou de publicar no Blood Advances, e realmente essa leucemia, geralmente 50% dos pacientes que entram em processo de relapse, desculpa, não lembro agora em português, mas que a doença volta, 50% desses pacientes vão ter uma superexpressão dessas bombas de transporte de substâncias, e uma delas é MDR1, que é a Multidrug Resistant Gene 1. E uma das substâncias que é usada atualmente na clínica, a daunorubicin e a idarubicin, são substratos para essas bombas. Então, eles acreditam que você trata o paciente e daí você vai selecionar por clones da doença que superexpressam essas bombas e a partir desse momento você não consegue reter essas drogas mais na célula cancerígena e essas células agora crescem e volta a doença. Essa é uma hipótese. Não são todos os pacientes que apresentam essas bombas, mas elas são realmente mais superexpressas nesses pacientes que entram né, que voltam a ter a doença. E a gente observou que o SMAC Mimetics é uma dessas drogas que também é substrato para essas bombas. Então, você pode fazer essa combinação, e tem inibidores específicos que já estão em clinical trials para essas bombas. Então, se você associa os dois, você consegue matar a célula, mas a célula vai morrer por apoptose. Agora, tem outros fatores. As células cancerígenas também podem adquirir mutações nos genes da apoptose. Então, leucemias se sabe que elas apresentam altos níveis de MCL1, que é um membro da família BSL2, que é muito importante e antiapoptótica. Isso é uma coisa sábia. E agora eles já estão, né, gerando inibidores de MCL1. Então, se você tem fatores como esses acontecendo na célula, você não vai ter apoptose acontecendo, porque as quimioterapias vão fazer pela mitocôndria, o MC1 vai bloquear. Então, são vários fatores, né? A AML é uma doença muito complexa, porque ela é muito heterogênea, tem diversos subgrupos, e ela é associada com outros oncogenes, não só as translocações de MLL. Tem outros oncogenes RAS, e outros oncogenes que também influenciam a doença. Então, assim, a apoptose pode ser descontrolada nessas leucemias de diversas formas. Então, se você pensa num sistema em que você pode começar a matar células com o sistema de quimioterapia normal, que seria Ara-C, daunorubicin, você elimina elas, e aí, se você pensar que você pode depois vir com uma necroptose que vai estimular o sistema imune para eliminar qualquer outra leucemia, célula da leucemia que ainda está no sistema, você pode prorrogar o survival, né, a sobrevivência do paciente. Mas, claro, isso é tudo, né, basic science. Quando a gente publicou esse artigo, a gente teve muito interesse das empresas farmacêuticas, tanto da empresa que produz o IDUM, né, o Emrixan, porque eles eram FDA approved, então você, teoricamente, poderia usar num clinical trial, mas a ideia de inibir caspases para tratar câncer ainda é uma coisa que as pessoas ficam meio tensas. Então, quanto mais a gente entende sobre a via de sinalização de necroptoses e como isso é controlado no câncer, essa ansiedade vai diminuindo e vai sendo mais, né, mais viável de se trabalhar. O nosso trabalho foi um dos primeiros trabalhos que mostrou que em vivo isso é uma coisa possível, e toda vez que eu apresentava, as pessoas falavam, mas você não tem um processo inflamatório? Eu falei, não, eu fiz tratamentos de biosafety em animais que não tinham leucemia, e eles receberam por 4 semanas, e depois foram analisados até 2 meses depois, e não havia nenhum problema, nenhuma sequela no animal. Mas até ir para o paciente, né, é um processo mais demorado, mas é uma coisa que é para se pensar, né?
Imaginei que assim, injeção de caspase 8, imaginei que todo mundo ficasse nervoso.
Esse é o key, porque não é todas as caspases, você não precisa inibir todas as caspases, você precisa inibir caspase 8 e só por um tempo determinado, porque a necroptose acontece em 4, 5 horas, essas células já estão destruídas, e elas não retornam, então é muito rápido. Então, não é um processo que você precisa continuar fazendo, e como eu mostrei, como você elimina muito rápido, não tem resquício de células que sobrevivem tanto. A não ser, claro, que elas tenham, como eu mostrei em pacientes que têm mutações na via de necroptose. Então, por isso que a gente está tentando entender isso um pouco mais, para identificar esses fatores de resistência.
E a minha terceira e última pergunta, né, eu fiz mais 5, mas depois eu mando para você por e-mail, é se existe algum teste no momento utilizando oncólise viral para esse tipo de leucemia que é tão resistente. Como você mostrou no plot, né, no Kaplan-Meier, você vê o desvio da morte mais para a direita, você tem um alongamento, mas no final ele acaba, claro, que aumentar a sobrevivência, né, quando se lida com câncer, o foco, na verdade, a gente obviamente quer curar, mas a gente vai passinho em passinho. Então, se você conseguir aumentar a sobrevivência, né, para 1 ano, para mais 2, para mais 3, e você vai chegando lá. E a oncólise viral, você viu algum estudo sobre isso, sobre oncólise viral em leucemia?
Oncólise viral, você diz...
O uso de vírus modificados para entrar nos tumores e se replicar no tumor e matar o tumor.
Isso é uma das tecnologias umas tecnologias, o meu inglês, meu português, é 1h da manhã, que está sendo sempre estudado, né? Eu acho que o que é complicado, principalmente com leucemia, é colocar vírus em células do sangue e ter certeza que esse vírus não vai ser reconhecido como um antígeno e vai ser eliminado de forma geral, e que o vírus só é localizado na célula cancerígena. Eu acho que isso é o mais importante. Então, há coisas acontecendo nesse sentido, mas o progresso ainda, né, que nem o CRISPR, é uma coisa que as pessoas estão pensando já. Mas o problema é que nunca se sabe o efeito a longo termo, né, com esses vírus inseridos nas células. E tem que ser um vírus muito eficaz em eliminar todas as células e que o vírus não se propague e gere outras mutações ou outros problemas. Eu acho que esse é o problema. Com relação ao nosso, que a gente prorrogou a sobrevivência, mas não eliminou, eu esqueci de falar, mas eu só tratei esses camundongos por 4 semanas. Então, só até o dia 30, daí eu parei o tratamento. Eu não sei se eu tivesse tratado por mais 2 semanas, por 6 semanas, se eu teria eliminado completamente, ou se eu tivesse... Quimioterapia, eles fazem, né, em blocos. Então, se eu tivesse feito 4 semanas, dado um tempo e voltado depois de 4 semanas, provavelmente eu ainda teria uma sobrevivência maior, mas a gente não fez isso porque por questões éticas de animais também a gente não podia fazer. Mas, então, tem toda essa optimização do tratamento que precisa ser levado em consideração. Mas ali, pelo menos a princípio, há uma indicação que esse tratamento pode beneficiar as células principalmente aquelas que são resistentes à apoptose. Eu não digo que nunca esse tratamento vai ser uma coisa que vai substituir o tratamento que já está em andamento no momento, que é super agressivo, mas que elimina as células. Mas é uma coisa que pode trabalhar com aqueles pacientes que voltam a ter a leucemia ou que não respondem ao tratamento de indução da apoptose, né?
Eu fiquei impressionada com o resultado, quando você mostrou o resultado in vitro, da redução, eu fiquei, uou, mas em vivo, quando você chega no paciente, aí você dá de cara com o paciente número 10, que não responde a nada. Meu Deus do céu, o que é isso, né?
Porque célula de paciente é muito diferente, né? É diferente, é outro mundo. Então, a gente faz tudo isso, mas aí você tem que acabar indo no paciente para ver como funciona. Mas ainda assim, 50% dos pacientes que a gente testou respondeu, o que é uma indicação boa, mas ainda tem coisas a serem resolvidas, né?
Sempre tem. Tá bom, então, Gabi, queria agradecer novamente a sua presença aqui no nosso seminário. Tem aqui várias pessoas que fizeram coisas bacanas, que gostaram muito da sua apresentação. Deixa eu clicar aqui. E, enfim, até a nossa próxima oportunidade de ver o progresso aí do seu trabalho. Tá bom? Então, se alguém tiver mais alguma pergunta, qualquer coisa eu posso mandar para a Gabi depois, se ela quiser.
Se alguém tiver qualquer coisa mais específica, pode dar meu e-mail, as pessoas podem me escrever. Eu posso mandar o artigo para quem, mas eu acho que vocês devem ter acesso aí. E a gente tem outras coisas em andamento também, né? Com o tempo, não queria só focar numa historinha.
Inclusive, até agradecer bastante a Gabi, porque ela está dando essa palestra na Austrália a 1h da manhã, né? Agora, eu pedi para subir um pouco para 10 horas, porque ela iria começar a 1h da manhã. O cérebro começa
Agora são 1h da manhã. Eu tomei um café antes, então tô bem ainda, mas ao meu olho, está começando, né?
Vai ficar pequenininho. Tá bom, gente, então,
obrigada, Joana, pelo convite. Né? Fiquem bem. Keep safe, as we say, com o coronavírus. E até a próxima.
Até a próxima, gente. Obrigada pela presença de todo mundo. Tchau.
Obrigada, Jana. Tchau.